Lost your touch.
Okay, so we are on chapter 23. Chapter 23 is about biotechnology. If you look at these pictures here, this is pretty cool. This is a glowing tobacco plant.
What? I want to grow that.
These are glowing fish.
Don't they have glowing
How do you have a glowing tobacco plant and a glowing fish? Tobacco plants don't glow.
Except under UV light.
Well, what they've done is they've taken a gene from a jellyfish, cut it out of the jellyfish genome, moved it into a tobacco embryo, a tobacco zygote, and it grew into a glowing tobacco plant that glowed just like the jellyfish does.
Do they have like really tiny scissors?
That's what we're going to talk about today, how they do it. And they do. They're called restriction enzymes, DNA scissors that will cut DNA. And so this is called genetic engineering. And genetic engineering is a big business, huge business. You may want to be a genetic engineer when you grow up, make a lot of money. Yeah.
So does biotechnology mean the same thing, or is that different?
Biotechnology, genetic engineering is a type of biotechnology. So we're going to learn about all kinds of biotechnologies.
Isn't this like one of the? Isn't this a really controversial science? It's basically playing God.
A little bit.
Yeah, there's some controversy about it, but I think if you learn about it, it won't be as controversial.
Couldn't you create a disease, like basically the plague?
Could maybe do that, yeah.
Wait, so we could develop a war with this, like you could make it a disease.
Maybe if you do bad with it, but you could also do good with it.
So could you make you can make your own like giant person?
What?
Well, maybe someday, but there's problems with being big that limit human growth, like circulation. It's hard to circulate blood around a very large body. The heart can only be so strong, and the vessels can only hold so much pressure. So that's a problem you'd have to somehow figure out. But they are using genetic engineering for a lot of things. For instance, they have corn plants where they've taken a gene from a plant that makes its own insecticide. And they've moved that gene into corn plants, and now the corn plants produce their own insecticide. And how is that good? Well, you don't have to spray your corn with insecticides anymore if you're the farmers that use this type of plant. And you know those insecticides are expensive. And whenever it rains, they run off into the oceans and cause all kinds of problems, killing things in the oceans and disrupting the food chain. And so now they have it where the corn just grows its own insecticide, and it makes corn a lot cheaper. And you can feed a lot more people, and there's less starvation already because they're doing this. Yes?
Isn't that the corn that they used to make plastics out of?
I don't know. Maybe.
Yeah.
Plastics are made from
Starching.
are made from molecules that are organic molecules. Plastic is derived from oil, which are derived from organic molecules. So, growing corn, you can make those molecules too. Yeah.
Oh my god. It's okay.
Would it like affect the taste on the video?
I know.
No.
I know that the plastic.
It's not.
The corn that like plastic, you can't even.
I am never signing up for this
Well, that's
again.
probably a different one.
It's okay.
Yeah, just try and be careful not to hit it because it's.
I am
it won't
so
stay on the
sorry.
thing. That's okay. So let's talk about some of the different types of biotechnology that they can do nowadays. One of them is called cloning. Have you ever heard of cloning?
Yes, sir.
Asa, you with me up here? What's going on over there? Is that the book or is that something else? Nice, it's the book. Do it, Asa. Cloning is a way of making new organisms and making them as identical copies to other organisms. How is this done? Well, the first thing that was ever cloned was a sheep. They call it Dolly. You ever heard of Dolly?
Mm-hmm.
What you can do is you can take a cell out of an organism, and they take this what they call the nuclear donor, and they pull a cell from the body of the nuclear donor, and they pull the nucleus using a very tiny needle. They inject it into the cell, and they pull the nucleus out of the cell of the nuclear donor. And then they take another cell, an egg cell, from what's called the cytoplasmic donor. And they remove the nucleus out of the egg cell, and they put this new nucleus inside of it. And then that will start growing into a new organism. They implant it in a surrogate mother, and she gives birth to the clone. And that is a clone of the organism that you took the original nucleus from. So they take the information of this organism, basically put it in an egg, and implant it into a mother, and she'll give birth to a clone of that organism. This would be possible to do with humans, but they don't do it with humans because it took 400 and some tries to get Dolly. All the other 434 to get a good Dolly, and the other 433 were all screwed up, malformed, didn't grow into a healthy organism. So you wouldn't want to try this with humans because they're not very good at it right now. Yeah.
So this removes the process of fertilization?
That's correct. That's right. Yep. You don't have to fertilize the egg. You can just take the nucleus from something else. So if we could clone humans and we wanted to make more Mr. Willises, we could just take one of my cells, take the nucleus out of it, put it into an egg cell, and implant that into a surrogate mother, and she would give birth to a baby Mr. Willis. And he would grow into an adult Mr. Willis. It would take however many years, 48 years to get as old as I am, but he would look like me going all the way up through. It's almost like an identical twin. It's the same DNA, and it would look the same. It wouldn't think the same unless it took all the same classes and read all the same books and was raised by the same people I was raised by. But it would be another person with my same genetic identity. Yeah.
What would happen to the extra chromosomes in the egg?
They remove them before they put the new nucleus in. Great question. They remove the extra chromosomes. So that's what they're showing here. They take that nucleus out with a little pipette there. Yeah.
So couldn't they do it? Couldn't they implant the zygote with the new nucleus in a test tube?
That's what they do. That's what they do. But it won't grow to a sheep in a test tube. You got to implant it into the uterus of a mother. So that's exactly what they do. Yeah.
Don't they they don't live as long as?
Well, Dolly didn't quite live as long as the mother did, but I can't remember exactly what happened, but they don't think it was because of anything that they had done. They just think that it died earlier. I mean, if you have two identical twins, they're not necessarily won't live the same length. One might die earlier from some kind of disease or something like that. So yeah, Dolly did die earlier, but they don't think it was anything from the genetic engineering. They've done this with all sorts of creatures, not just sheep. They've done it with rats and dogs and cats and horses and cows. All types of different things have been cloned now. And it's real good for scientific experimentation. If you want to remove a variable from your experiments, if you're doing, say, experiments on mice, make all the mice the same. And then that's one variable removed. You don't have different mice. They're all exactly the same. So if you give this one a drug and have these mice a drug and these is your control group, the only difference between the two groups would be the drug, not the fact that the mice are different themselves, because the mice are all the same because they're clones of one another. So they like to use clones in scientific experiments.
I forgot my question.
Oh, I'm sorry, I waited too long.
If they took a nucleus from the clone,
Yes.
to make another clone,
Cloning a clone.
would the DNA eventually degenerate?
Probably not, since it grows up into a healthy, as long as it's a healthy adult, it should be just fine. So, if you think about it, we've been copying the DNA to the next generation for 4 billion years now, some odd years, from generation to generation, and that maintains a working bit of DNA. So it'll probably be the same way here. So that's cloning. That's a type of biotechnology, and they started doing that when I was about your age. They came out with this dolly. I think it was 1990 or something like that. I don't remember, but we were like, "Whoa, that's pretty cool." And hopefully there'll be an army of Mr. Willises out there. Do you want to see the dolly, the actual sheep?
Yes, sir.
You know, they still have Einstein's brain. Maybe they could clone Einstein from some of this stuff.
That would be pretty cool.
And now we can have more Einsteins to solve all our tough problems.
I think he was smart and.
In 1997, Dolly became the first mammal ever to be cloned artificially from another adult sheep, proving once and for all that even adult cells contain the necessary information for creating each part of an entire organism. Let's see how. Using microsurgery, scientists first removed the nucleus from an unfertilized egg of an adult sheep. A complete udder cell was then taken from a 6-year-old ewe. The nucleus from this cell was inserted into the emptied unfertilized egg.
So they just injected the nucleus into this egg. And now this egg cell has the nucleus from that other sheep in it, and this will grow into a new organism that has the DNA from that other sheep. How cool is that?
Yes.
Really cool.
The egg now contained a complete set of genes from the adult udder cell. After implantation into another sheep, it grew into a complete new individual. Dolly is an exact genetic copy, a clone, of the sheep that donated the udder cells.
In creating Dolly, the scientists...
Okay, that's about it. She just starts talking about other stuff here.
Yeah.
Yeah. What's that?
Kind of creepy.
Doors open.
Those are the ghosts that live here. Y'all don't know about the ghosts?
Oh yeah, they just randomly open when you're watching the video.
All right. This building is on an old Indian burial ground. Did y'all know that?
Mm-hmm.
No, I did not know
True
that.
story. They moved all the bodies, but there used to be
No, but it's not
Indian
as
cemetery
creepy.
here.
I had a really good question.
This DNA, remember, has A's and T's and C's and G's. Y'all remember that? Now, there's been a project that started also in the 1990s called the Human Genome Project. That you should know about that, the Human Genome Project, where they went through, they have ways where they can determine the A's and T's and C's and G's that make up DNA. And they went through one by one and figured out each letter in order. And they got DNA from several different people around the world, and they figured out all the A's and T's and C's and G's of all these people, and they compared them to one another, and we call that the Human Genome Project. And it's 3.5 billion letters. 3.5 billion A's, T's, C's, and G's in each cell that make up the entire genetic code. And they have all that down in a computer. You can look it up online and see all the A's and T's and C's and G's. And what they're doing now with that information is they're going through it, trying to figure out what makes people different. What are the letters that make people different? Why do you look different from me? Because you have different code than I do. But we have a lot of the same code. As a matter of fact, for any two humans, their codes are 99% identical. So it's only 1% of the code that makes you different from somebody else. And what they're trying to do is go through that code and see what makes people different, what genes make people different. Like, obviously, some people have a gene for making dark hair, and some people have a gene for making light hair. And they're trying to go through and find out where the hair color gene is. And then, if they figure out what letter combination makes dark hair and what letter combination makes light hair, then they could take maybe DNA that they find somewhere, and they could read it and see if that DNA comes from a light hair person or a dark hair person. And once they know the whole code, they'll be able to plug it into a computer and that computer can draw a picture of what the person should look like just from a DNA sample. Wouldn't that be helpful for solving crimes?
Mm-hmm.
I think William was first.
I remember my question.
Okay?
So for clones, would the environment have an effect on how they were, like, how they look? And stuff?
Absolutely. Just like two identical twins. If one eats a lot and one doesn't eat a lot, that identical twin is going to be bigger.
So
the environment does have an effect.
even if it's a clone, it can still look completely different.
It could look different. It's not going to look completely different because a lot of your genetics determine how you look. But just like two identical twins can look different. One can cut her hair and one can grow long hair. One can lay out in the sun and one can not. You'll look a little different because of your environment. Yes?
So how come when people like start getting older, their hair starts changing color?
It becomes gray, they think, because these little peroxisomes. Do you remember the little bubbles in cells that hold hydrogen peroxide? They think that those things will start bursting and causing the hair to bleach white. And they're trying to figure out a way around that. But that's what happens when you get older. Your hair becomes bleached white. They're not sure how to fix that, but maybe they can one day, and then nobody will have gray hair. Wouldn't that be great? Yeah.
So, two things. Would they be able to like cut and snip what they don't like in some of these genes?
That's what they're learning. That's what they're learning how to do. Now, here's the thing. All of your cells came from an original cell. So if they want to change the DNA in all of your cells, they got to change it in that first cell. Because after it multiplies into your 80 billion cells, how are you going to change it in every cell now?
You won't be able to.
It'd be hard, wouldn't it? It'd be hard to go into every cell and remove this piece and put in that piece. So for genetic diseases like Huntington's disease or cystic fibrosis or something like that, you might have to catch it really early and change the earliest cells, and then they become all the other cells. But that's what they're trying to figure out, how to take out letters that are from mutations and put in letters that are normal to get rid of some of these genetic diseases. That's what people are figuring out right now. This is what you guys hopefully will figure out. You go to college, you major in biology, major in genetic engineering. I think Georgia Tech has a good genetic engineering program. People who come out of school with those degrees make $100,000 a year right out of school. I mean, this is big stuff. So if you're into this kind of thing, it's a good job to get into. Yeah.
And they have been able to identify the face from DNA. There's this National Geographic issue that said this is what its DNA is, but he might be heavy set or thin.
Oh yeah.
So they can already do that.
You can send your DNA to something.
Yeah.
There's some like article in New York. This lady picked up a bunch of like random things, like a cigarette butt or a cup, and took the DNA off of it and manufactured a face of what it would look like.
Using the
It's not
DNA.
necessarily the person.
Yeah.
Yeah, but it would only be like their facial features.
Yeah, now see, that's new. They didn't used to could do that, but they're understanding the genes better, and one day they'll be able to know all about a person just from their DNA, and that'll help you. Right now, you can send it in, and they can tell you, send your DNA in, and they can tell you, hey, you have a greater chance of getting breast cancer, or you have a greater chance of getting prostate cancer, and that sort of thing. And that might be good to know. If you have a greater chance of getting some sort of cancer, you may be able to change your diet or exercise or do things that will reduce your chance of getting that particular type of cancer. So there's already things you can do with your DNA by you spit on something and you put it in a bottle and you send it in, and they'll check your DNA for you.
So when you do like the family, sometimes you can
Family tree stuff.
do like the family tree stuff with your DNA. How can they tell you're this percent this, that?
Yeah, they
that?
take your DNA and they look for genes. There are genes that are just in Europeans, and there are genes that are found in Africans. And they check for those genes with these tests, and they can tell you what percentage you are of European or African or Asian or whatever. So this is interesting stuff, the Human Genome Project. Now, let me show you about what's called recombinant DNA technology. So, 10 points if you can remember this. there is a loop of DNA that's found in bacteria. A loop of DNA.
Not a helix.
And it's not the chromosome of the bacteria, but I don't know if you remember this, but we talked about how they could pass it from one bacteria to another through a little sex pilus. They could take a little loop of DNA and pass it through the sex pilus, and it's like bacteria sex. If you can tell me what that loop of DNA is called that can be passed through the pilus, I'll give you 10 points.
So they don't have helixes, they have
loops. Orange, correct, but it is a helix. It's a double helix. It's just in a loop.
It doesn't look like a chromosome.
Right, it's in a loop. Blue, correct. Peach, correct. White, correct. Purple, correct. Anyone else?
And it would probably be higher than that.
Yeah, we're coming.
Wait.
Now, William, their main chromosome is a real big loop, and then these things are little tiny loops. So, yeah, but it's always in a loop in bacteria. No one? It's called a plasmid. A plasmid is a loop of DNA. Now, I'm going to show you what our scientists have already done. This represents a plasmid. Listen, this thing... William, could you turn the light on for me?
Yes, sir.
I'm going to do this on the board. And you'll have to show this over here with the camera. So, a loop of DNA is a plasmid. And what scientists have done is they're able to remove plasmids from a bacteria. And they're able to cut the DNA with special DNA scissors. This is an enzyme that's a form of DNA scissors. And these enzymes are called restriction enzymes. Can you say restriction enzyme?
Restriction enzyme.
Louder and at the same time?
Restriction enzyme.
A restriction enzyme is a special enzyme that cuts DNA. And this one that scientists like to use, there's a bunch of them that they use, but there's one that they talk about in the book. It's called EcoR1. And it was found in the E. coli bacterium. And what it does is it cuts DNA wherever it sees the sequence GAA TTC. Wherever this enzyme reads GAA TTC, it'll cut the DNA. GAA TTC is a palindrome. You know what a palindrome is? Something that's read the same forward and backward. Like race car. Did y'all know race car is a palindrome?
Mm-hmm. And my name.
Race car reads forward and backward.
And evil.
ASA
Evil?
Or live.
is a palindrome.
Yeah, live.
ASA.
No, what is that called? What's it called?
Now, listen. GAA-TTC is a palindrome of sorts because it reads GAA-TTC on one side, up here, and on the other side it also reads GAA-TTC going the other way. And so whenever EcoR1 sees GAA-TTC, it cuts between the G and the A. And so here's our plasmid that we got from the bacterium. And we can put this plasmid in a test tube and put this enzyme in the test tube with it, EcoR1, and wherever it reads GAA-TTC, it'll make a cut right there between the G and the A. And it cuts on the other side too, because it reads the other side backwards. GAA-TTC, and it'll cut there, and this thing will open up like that. And it'll come open. And then what the scientists can do is they can take human DNA, for instance, or spider DNA, or corn DNA, or DNA from any organism that they find, and they can use the same restriction enzyme to cut that DNA up into pieces. So if you notice, this piece of DNA has 2 of these GAATTC strands. One right here, snip, snip, open up. And another GAATTC down here, snip, snip, open up. And what they've done is they've cut out this piece of DNA. Hold on just a second. And then they can transfer that DNA into the test tube, and it will join with the other DNA that's cut, because there are these jagged ends that scientists call sticky ends that allow the A and the T and the T and the A to bond together. And they need another enzyme called DNA ligase to come in here and put the DNA in to glue it in. And now we have a plasmid that has extra DNA glued in. This is how they made the tobacco plant glow. They cut the gene out of the jellyfish and they glued it into the tobacco plant. And now the tobacco plant has the jellyfish gene for glowing. Yeah, what were you going to say?
So do they just take like the restriction enzyme from E. coli?
Exactly. They take these E. coli bacteria, and the E. coli bacteria have a lot of the restriction enzymes in their little cells. And it's to protect the bacteria from viruses. This enzyme will cut up virus DNA and protect the bacteria. But what the scientists do is they open up, they put chemicals that kind of destroys the bacterial cells, and these enzymes flow out, and they have a way of separating these out and getting them, and then they can put them in test tubes to cut up DNA with.
How do they put it? Do they put it in a cell so that it can go to the DNA and cut it up?
Well, no. What they'll do is they'll take cells out of some organism. Like for me, they could take cells from my hair, or cells from my skin, or saliva cells or something, and then put those in a test tube, and put the restriction enzyme in the test tube, and it'll cut it up right there in the test tube.
What's the piece that is called?
This piece right here, this is a gene for something that has code for making something.
Does it have a special name?
Well, it's just a gene. And you can insert the gene into a plasmid, and now this plasmid can be inserted into a bacterium. And the bacterium will now read the code and make the protein that this code's for. Let me tell you a way that they're trying to do this. What they're trying to do. Check this out. This could change the world. Think about this now. They take spider DNA. And somewhere in the spider DNA is code for making spider silk. Did y'all know spider silk's some of the strongest stuff on Earth?
Is it stronger?
Y'all heard that. Stronger than steel. Much stronger than steel. Stronger than Kevlar. And they can cut the gene out. Listen. They can cut the gene. They can cut the gene out of the spider DNA. They can paste it into a bacterial plasmid. They can put the plasmid into a bacterium, and the bacterium will start making spiderweb silk. And they can feed the bacteria so it reproduces itself. And goes from 1 bacteria to 2 to 4, 8, 16, 32, 64, 128, 256, 512, 1024, 2048, 4096, 8,000, 16,000, 32,000, 64,000, 128,000, 256,000, 500,000, 1 million, 2 million, 4 million, 8 million. You got this whole big, they can grow these huge vats of bacterium, and every one of them is a clone of the original, and every one of them can make spiderweb silk. And the spiderweb silk comes oozing out of the bacteria's body, and you simply strain it. You have a strainer that strains out all this protein, and now you got a bunch of spiderweb silk. And you can weave that into clothes, and guess what? Spiderweb silk is so strong that if this shirt was made of spiderweb silk, it would be bulletproof. And think about how that would change the world if shirts could be bulletproof. Think who you could sell to. Police, Army. Heck, I'd buy a bulletproof shirt and feel a little bit better walking down the street, wouldn't you? Mm-hmm. And what if you line tanks with this stuff? I said it's stronger than steel. Think how that would change what a tank looks like and how fast it can go and that sort of thing.
Wait, what's stopping them from doing
Think how it would protect your car. What's stopping them is they can't do it very well right now. They can cut this this out and they can put it in bacteria, but the bacteria don't produce enough of it. The proteins get tangled up inside the bacteria. They're trying to do this right now, though. Maybe they'll have it one day and they'll figure it out. Yeah.
Didn't they do this with goat's milk?
They did. They've got it where goat's milk will produce spiderweb material. They put the gene into a goat, and the goat produces the spiderweb material in its milk. And they're trying to get it that way. But it's not just I mean, that's just one tiny thing that they could do with this. They've already taken, did I tell you about the insecticide?
Yes, sir.
They've already taken gene for insecticide and put it in corn, and now they're growing corn and they don't need to spray insecticide on it. They've taken the gene, is anyone here diabetic? If you have diabetes, you have to inject yourself with insulin. All the insulin nowadays, not all of it, but a lot of it, is made from bacteria that have a human insulin gene inserted. And the bacteria produce human insulin.
So why don't we put this bacteria in our body?
Because that would cause an infection that would kill you.
Yeah,
You don't want the bacteria in there, you want the protein.
Did they find a bacteria that doesn't kill you, like the ones in yogurt?
Yeah.
Maybe. That might be the next
Yeah, what
thing.
if they have
Become a
insulin?
genetic engineer and work on that.
What about
and get it done.
Insulin yogurt. Like you just eat the yogurt.
Just eat the yogurt. Here's the problem with that is your digestive system breaks down proteins. So you can't just eat the insulin. It'll be digested.
You inject yourself with yogurt.
You have to inject yourself. Yeah.
So
Oh, we're out of time. Make sure you do your reading.